Well, good morning, everyone. I'm Rick Lifton, and I'm the president of Rockefeller University, and I'm really delighted that so many of you have come out this morning on this chilly, damp morning to be with us for this Women in Science program. Before I introduce today's speaker, I'd like to say just a few words about Rockefeller University for those of you who are new to campus. The university was founded in 1901 by John D. Rockefeller, who, with his vast wealth, wondering what would be the best use of it, realized that we understood almost nothing about the fundamental causes of human disease, and that without that, all of the treatments that were being used were very unlikely to be of any value to patients who were getting them. So he established the Rockefeller Research Institute, now the university, with the goal of understanding the fundamental causes of disease, with the expectation that this is going to provide the best opportunity to develop new and meaningful ways to diagnose, prevent, and treat disease. And that has led to our credo, Science for the Benefit of Humanity, which has been a consistent effort here for the last 125 years. So our laboratories have conducted basic biological research in this direction, and some of the most important discoveries in the history of biology have occurred here on campus. Among these, none more important than 1944's discovery of DNA as the genetic material. And this transformed our understanding and the ongoing research in biology, biotechnology, and pharmaceuticals that has dramatically changed the public health. So the very small environment here, we only have 70 faculty at one time, enables our scientists and graduate students to commingle with one another. It's an extremely cohesive community. People from different disciplines interact with one another at the highest levels, and it makes for a spectacular environment on campus. The success of the institution has been nothing short of remarkable in our history. There have only been 270 professors in our history, and of those, 26 have won Nobel Prizes in medicine or chemistry, and no other institution anywhere in the world has a percentage anywhere close to that number. In addition to Nobel Prizes, Lasker Awards are the U.S. Nobel Prize, and we've also received 26 of these. And most recently, just this September, Svetlana Moisov received the Lasker DeBakey Clinical Medical Research Award, and that was our 26th Lasker awardee and the first woman on the faculty at Rockefeller to receive a Lasker Prize, and we're extremely proud of that. And Svetlana, I think, is here. Svetlana, please stand and be recognized. Congratulations. Now, there are awards and awards, but Svetlana's award is a remarkable one because it's for her discovery in 1988 of GLP-1. Many of you who don't know a lot about science may nonetheless be familiar with GLP-1 because it's the foundation of the drugs that cause profound weight loss as well as effective treatment of diabetes. So Ozempic, Wegovy, Mounjaro, and Zepbound are all based on the technology that on Svetlana's remarkable discovery. So our program today is presented by Women in Science, and this is an initiative that was founded more than 25 years ago to support women researchers like Svetlana who receive exceptional training while here and then go on to distinctive careers. So since this initiative was started, more than 270 of our graduate students and women's graduate students and postdocs have been awarded coveted Women in Science fellowships. Some of these women are and science fellows are here with us today. Would you all please raise your hands if you're here? Anyone? Well, Svetlana could raise her hand. It's early for scientists, you have to realize. So Women in Science has also helped us to recruit women to the faculty and has provided funds for the expansion of the on-campus Child and Family Center and most recently established the Anna Maria and Stephen Kellen Women in Science Entrepreneurship Fund to encourage more women to realize the therapeutic potential of their research. So you can learn more about the achievements of women in science in your program. If you're not already involved with the initiative, I urge you to join this amazing community of women who are passionate about learning how basic science is leading to better health outcomes. So it's now my great pleasure to introduce to you our speaker this morning, Elizabeth Campbell, who is a longtime member of our academic community, who also happens to be Rockefeller's newest tenured professor. Liz is a brilliant investigator whose research on the fundamental biology of microorganisms is helping medical science to envision improved treatments and cures for tuberculosis and coronavirus infections, two of the greatest current threats to public health. Tuberculosis is an ancient disease that's caused by the bacterium, Mycobacterium tuberculosis, and it is the single largest cause of death from infectious disease on the planet, eclipsed only briefly by COVID-19, which, as you know, emerged about 5 years ago as a previously unknown coronavirus that took hold in the human population and caused a very large number of deaths worldwide. These pathogens are completely different, but Liz has made fundamental contributions to our understanding of these microorganisms and strategies for how to treat them. So using an astonishing array of technologies ranging from basic biochemistry to cryo-electron microscopy of protein structure, Liz is making fundamental contributions to the understanding, in particular, of how the genomes of these microorganisms are expressed. Her investigations have provided us with new ways to see how these infections begin and progress. Developing drugs for TB is a really critical issue. As you may know, because of its prevalence, existing antibiotics, resistance has emerged to virtually all of them, and now the treatment of this disease is extremely complicated by the high degree of resistance in the population, some to all of the existing antibiotics, and it's recognized that developing new antibiotics for TB is a critical public health effort. Developing antiviral drugs is a completely different kind of challenge, and Liz has made vital contributions to this field as well. So for her contributions to all of this work, Liz has been extremely well recognized by the microbiology community and beyond. And in 2023, Liz won the Emil von Behring Prize from Marburg University, which recognizes outstanding achievements in the fields of immunology, microbiology, or virology. This prize has been given every other year since 1942, and Liz is only the second woman to receive this honor since that prize began, and we're very proud that she has received this recognition. In fact, the only other Rockefeller researcher to win this award was Ralph Steinman, who won the Nobel Prize here at Rockefeller in 1996. So Liz has a fascinating history. She was born in Guyana and moved with her family to Queens when she was 12 years old. She went to Swarthmore as an undergraduate, where she first became captivated by the field of microbiology. Her interest brought her to Rockefeller for her graduate study under the tutelage of infectious disease researchers Elaine Tumanen and Robert Missouri. And as a student, she studied how pieces of DNA are transferred from one bacterium to another. And from this is how bacteria rapidly can transfer bacterial resistance from one bug to another. After graduation, she stayed at Rockefeller as a postdoctoral fellow in the laboratory of Seth Darst. And there she became expert in structural biology and studied mechanisms of transcription and how transcription initiation occurs. And she went on and stayed there. And about 15 years ago, Liz turned to tuberculosis as a field, recognizing that the studies that she had been doing in the Darst lab on the commonly used bacterium E. coli, which has been kind of the gold standard for everyone to use for studies of bacterial transcription in bacteria, we didn't really know very much about how tuberculosis transcription works, but everybody had assumed that it's pretty much the same everywhere. And this turned out to be wildly incorrect, as Liz showed by her pioneering studies of transcription in TB. And in fact, there are many elements of it that are completely unique. And that may not be so surprising because tuberculosis and E. coli last shared a common ancestor about 1 billion years ago. So this was one of those areas of science where you see her work and you think, why didn't anybody think of this a long time before Liz? But this is why science is so much fun, because somebody with a unique idea can have tremendous impact. So she has continued to look at how transcription is initiated, and this turns out to be one of the most important targets for antibiotic effects, and is a very strong target to be working in. So for her prolific and impactful research at Rockefeller, she was promoted to tenured professor and head of the Laboratory of Molecular Pathogenesis this last June. She chose the lab name because it was the name of the Rockefeller lab she joined as a graduate student with Drs. Trumanin and Missouri. So Liz's team is moving in the former lab of another Rockefeller luminary, the late Mary Jean Creek, who was a pioneer in research on addiction. Mary Jean's career has been especially inspirational to women scientists, and Liz, I'm sure, looks forward to honoring Dr. Creek's legacy in this historic space on the Rockefeller campus. Liz's promotion carries with it another important distinction. She was recently named the Corinne P. Greenberg Women in Science Professor. This new professorship was supported by the Greenberg family in honor of the late Corinne Greenberg, who served as a founding supporter of women in science and has been a generous benefactor to the Rockefeller University. Liz's contribution to science extend well beyond the bench. She teaches in the Biochemical and Biophysical Methods course and serves on the Tri-Institutional MD Ph.D. Admissions Committee. She participates as well in the Rockefeller Inclusive Science Initiative and actively mentors aspiring investigators, particularly women and underrepresented minorities at all levels in academic science. So this will be Liz's first Women in Science talk, and I'm absolutely delighted to join you in welcoming Liz Campbell to the stage.
Good morning, everyone. Thank you for coming in so early and, like Rick says, braving the weather. I'm really honored to be here and to give this lecture, and I'm also really honored to, as Rick says, be a professor here at Rockefeller University. So what I'd like to do today is tell you a bit about my program, what we've done, what we're doing, and how we're thinking about moving forward into the field of addressing pathogens. So before I start, I would like to acknowledge the people that support and the institutes that support our research. I'm really lucky that I have a lot of internal grants here. I'm supported by the Rockefeller Ph.D. program, for example. The SNF is a big supporter of my clinical work. And I also use the cryo-EM centers here at Rockefeller, the Evelyn Grasslipper cryo-EM Resource Center, but also I use multiple resources here as well, including, you'll see some examples of the Precision Technology Center as well. So Rick gave you a little bit of my background, but I started doing this just because I've been asked to do this at other universities. I'm giving you a little bit of a background about who I am. I was born in Guyana in South America, and I did come here in 1982 to Queens. And I like to show this because I often, people get a little perplexed when they hear my name is Elizabeth Campbell. Where does that come from? This is my 23andMe. So in Guyana, in Guyana, I am actually a descendant of the indentured serfs and slaves that did the land, tilled the land. And also the Native Americans in the Caribbean, which are the Lakota Arawak Indians. But I also have European ancestry. So, for example, my grandfather was a Campbell, as well as Native American. So that's who I am. And what about my academic journey? So I went to Flushing High School, which was the first public high school in New York. And then I went to Swarthmore College, which was very transformative. This was a place that took the time to take someone from what was an underperforming high school and really did a lot of work to teach me how to think critically, how to write critically. And I am grateful for that, to that institute. And also grateful to Rockefeller, who also did the same thing. Really, really did a great job in terms of teaching me again how to think about science, how to write about science. And then I did my postdoc with Seth Dars in molecular biophysics. And then I took a very I took not necessarily a detour, but I took a unconventional path in a way. I took some time to raise my two daughters. Both of them are now graduated. One of them is in the audience right now. But at the same time, I was trying to run an independent program studying pathogens in Seth's lab. But in 2020, I started studying coronaviruses. And like Rick said, I did win the Von Behring Award, and that made me take pause and made me think maybe I should be doing something else with my life. So I went into the open search at Rockefeller University and was successful, I guess. And I was promoted to a full professor here in the laboratory. So the lab just started. We haven't even moved into our new space yet, but we're really excited. But I'll tell you some of the work that I've been working on in the background, and even like work that we've done this year with just, the benefits that come with being able to run your own lab. So the landscape of pathogens is vast. This is just a sampling of how different, bacteria are, how different viruses are, all the different pathogens. And so one of the things I've come to recognize is they're all different. So you actually need to study the biological systems, the biological processes within the pathogen themselves. And the two that I've chosen to work on is Mycobacterium tuberculosis and coronaviruses. And that's because they are two of the deadliest pathogens. And the bacterium that causes TB or tuberculosis is Mycobacterium tuberculosis. So I'm going to tell you a little bit about that. I sort of abbreviate it and say MTB sometimes, or TB. And then I also wanted to give a shout out to the people that do the work as well. I'm going to first start talking about the TB group. So, and many of them are here, over there somewhere. And so I'm really grateful to this group of really talented, hardworking people. And then later on in the seminar, I'll tell you a little bit more about our coronavirus projects that are co-directed with me, Seth, and Charlie. So what is TB? And this cartoon from the CDC basically tells you what the symptoms of the disease are. It's a debilitating disease. It's a lot of suffering is associated with it. You've probably seen movies of when TB was a problem. You're coughing up blood. You're weak. You have night sweats. You're shaking. You have no appetite, and often death was associated with that. So what I'm showing you here is a bit of the pathology of the disease. It's mostly pulmonary, so it occurs in the lung. It's transmitted by aerosol, and then they take residence in the lung. They hide in your immune cells. So they hide, for example, in these little nodules that we call granulomas, and they're mostly non-replicating at that point. So they're hiding out in your immune cells, and which is a bit ironic because the immune cells are supposed to be clearing the infection. But they can also go from latent. So latent TB is dormant. They're not really causing, there are no symptoms. But if they become active and they start to replicate, they will destroy your lung. And that's active TB. And you can see that in pink. They're spreading. They're destroying parts of the lungs. And then part of that destruction is they leave a hole. The lung can collapse. There's a cavity. And then when that happens, it's pretty bad. That's when the disease is at its worst. Part of active TB is that it can spread. It can leave the lung. And many people might not be aware of this, but it can be extrapulmonary and cause a disease known as Pott's disease. So it's estimated at least about 10% of TB cases can be extrapulmonary. And it can take up residence in almost any part of the human body, like the spine, for example, causes abscess. And you get this hallmark deformation of the spine here. And this is actually a picture from of a mummy that they think might have had Pott's disease. And this is something from the 1920s and 30s from a medical textbook. But unfortunately, right now, we do have extrapulmonary TB, and that's what it looks like in real life. So I'm going to give you a little bit of a historical walk here. It's early before we get into the science, so I just want to tell you some a little bit of information. So TB is an old disease. It's been found in Egyptian mummies, for example, and it's thought that Queen Nefertiti and her pharaoh also died from TB. But many of you might be aware of like a lot of luminaries in art and literature, in politics have actually been victims to TB. And this includes Eleanor Roosevelt, who died in TB right here on 74th Street at the age of 78. So she actually had a case of TB. It went latent. And then later on, I think she was ill, so they treated her with steroids that suppressed her immune system, and then it activated TB. So she actually died from disseminated TB. And as I mentioned, it is a disease of the suffering. It's taken a lot of lives very early before antibiotics. So John Keats died at the age of 25, longing for death, because as I said, it's a disease of suffering. And you might have seen, illustrations of artists who've also lost people to TB, like Edvard Munch lost his sister who was 14 years old, and Claude Monet lost his wife at the age of 32. But the good news is TB became treatable. So part of it, a big part of it actually, was education on increasing hygienic practice. Don't cough on people, cover your mouth, don't kiss your baby in this case. And then antibiotics came on the scene. So in the 1940s, streptomycin was discovered and to be used to be able to treat TB. But then quickly it was realized, because of this disease, the nature of this disease, it's latent, it's hiding in the lungs, that you actually needed combination therapy. You needed more than one antibiotic to treat this disease. So that's where we were with TB. We had antibiotics. So why are we talking about it in 2024? This is a graph from the World Health Organization, and it's just showing you the cause of death across the world. And you can see here TB is the leading cause of death by a single infectious agent. So that's what this is showing here. The rest are heart disease and diabetes and kidney. So also, it's worth noting that even though it's 1.3 million people dying a year of TB now, is that about 1.8 billion people in the world is estimated to be infected with latent TB, the one that's hiding out in your lung, and about 30 million of those people are in the U.S. So in 2020, coronavirus, like Rick mentioned, sort of took the title of being the leading cause of death by a single infectious agent. But unfortunately, TB is back. TB is back again to having that title. I will say, just in case any of you are interested in what are the risk factors for latent TB, is that some of the common treatments that are used to treat diseases such as rheumatoid arthritis or Crohn's, or steroids, for example, they can actually cause the reactivation of TB. So people who have latent TB, if you're tested positive, have to be treated with antibiotics to address their latent TB before any of this can happen. And there are other events such as organ transplant. You're also treated with immune suppressants. So that's where we are with TB. It is a disease of the impoverished, so many of us might not know all of these details. The reason is that it actually affects countries that are poorer. And so most of the impact occurs in these countries, in Asia and Africa. And unfortunately, about 400,000 active TB cases are now drug resistant. And as I mentioned, why is it a disease of the poor? It's because one of the biggest correlation with TB is access to nutrition. So the World Health Organization has done a very admirable job in addressing the global burden of TB. So they basically have been doing this for about 20-something years, and they go into these countries and they make sure people who are testing positive for TB are taking their antibiotics with direct observed therapy. And so they've estimated about 80 million lives have been saved. So this is something that's addressable with the right approach. And as I mentioned, it's a combination therapy. So already, if you don't have drug-resistant TB, the combination is about 4 to 5 antibiotics. It's 4 to 6 months. And this is what you're taking daily, and you have to take it, otherwise you're going to get drug-resistant TB. You need to finish your course. If you have drug-resistant TB, it's more dire. It's up to 2 years. And then there are a lot more pills that they're taking daily. So you can imagine that this is hard for compliance. One of the reasons is that these antibiotics are toxic. They have toxic effects. So like they can cause neurological diseases. They can cause blindness, deafness, etc. So it's a lot of work for the World Health Organization to ensure people are finishing their regimen. And why are antibiotics so critical for us? Maybe you already know this in the audience, but it's always striking to me to see how much death fell, right, on the advent of antibiotics. And it basically doubled the life expectancy once antibiotics came on the scene. But unfortunately, we got a little, relaxed in our approaches of thinking about antibiotic discovery. So between 1990 and maybe 2020, there was a discovery void. It's picked up a little bit now. And I just read this article in the New York Times that basically said one of the reasons is that it's not a great financial incentive for the pharmaceutical to develop this, because if a pill cures a disease, that's it, right? But so chronic diseases such as high cholesterol are more profitable. So a lot of this falls on on academia, on philanthropic efforts to really address this problem. There is now a new threat, bigger than even including not just TB. Antimicrobial resistance is a general threat. Lots of pathogens are resistant to antibiotics. And although they've revolutionized modern medicine, they're starting to reverse this trend. So before antibiotics, these are some of the statistics, right? Women died from childbirth, for example. Pneumonia was not curable. But now there are 2 million infections a year, and because of antibiotic resistance, there's about 23,000 deaths in the U.S. And it's estimated that in the next 25 years, that antimicrobial resistant pathogens are going to be the cause of death. So I would argue, and this is a good time for the talk, I guess, that maybe now is not the time for us to stop drug discovery. Now is the time for us to accelerate it. Now is the time for us to think about the future, be visionary, think into 2050. I'm hoping this won't be true, but yeah. So now I'm going to talk about my specific research here. So what are the challenges for developing new TB drugs? So we have to find the right targets. We want to find things that aren't going to cause those toxic side effects. We want to find the combination. The drugs have to interact with each other, can interact with each other. They have to, sort of work well together. And they have to be active against the non-replicating bacteria. So I'm going to argue that one of the reasons that we haven't been able to eradicate it is very hard to treat latent TB. And it's, people might not know they have it, but once they know they have it, it's also very hard to treat something that's not metabolically active. And then we have to get access to the infected population. So this means oral antibiotics, because we're not talking, access to, I'm really lucky I live next to Weill Cornell, so if anything happens, I just hop over there. But that's not necessarily the case for many people. And this is where I come in. So rifampicin is one of the first-line treatments for treating TB. And I think, and I think I'm supported by this, it is the most critical component. It's the only sterilizing component in the first-line treatment of 4 drugs. And it works by targeting an enzyme that is the love of my life, which is RNA polymerase. And RNA polymerase is a central enzyme of transcription. So now we're going to have to wake up and go a little bit into science. So what is transcription? And I apologize if you already know this. I know many of you will come to these seminars. But I just want to remind you, transcription it's taking the information that's stored in our genetic code in DNA, and it's transcribing it into an intermediate called RNA before proteins are made. And proteins are the building block of our cells. So this is the central dogma of molecular biology, and I'm interested in this section, right? Taking DNA and making RNA, and I'm going to talk about the RNA polymerase as a target for antibiotics. So this slide, you can glaze over it. It's just to demonstrate the complexity of what transcription is. It's not just making RNA. It's highly regulated. It has to go through multiple steps. The enzyme is not just synthesizing this, this molecule of RNA. it's doing a whole bunch of different things. And I'm going to try to address a little bit of that. But this is basically what I center my program around: understanding how this works, how is it regulated, what are the the fine details in what this enzyme is doing. And the reason I do that is because it's targetable. I told you rifampicin is a critical drug for treating TB, and it's targeting transcription. But there are lots of inhibitors that can target transcription, that can inhibit, that can prevent the synthesis of RNA, but they're not drugs. They're not antibiotics. We call them inhibitors in the lab. But what they can do is they can represent the next class of inhibitors, right? We can think about something that's working on this step, maybe of developing it as the next class of inhibitors against TB and maybe other pathogens. So this is where the talk is going to go now. Can we look at the RNA polymerase, at transcription again? And I'm going to tell you how we've been working on transcription in TB. So my questions that I'm going to try to address here a little bit today and just give you a sprinkling of how we've been doing this is I want to understand how antibiotics work. I want to know the exact step transcription they're inhibiting. I want to know why and how. I also want to understand something called intrinsic antibiotic resistance. So TB is one of these unique bugs that if you give it antibiotics like streptomycin, that anything that targets the ribosome, it will actually upregulate a bunch of pumps and just pump it out. So it has intrinsic. It's not a mutation. It's just they just have this intrinsic mechanism to pump it out. So we're trying to understand how this occurs. We also want to understand new mechanisms of resistance. Resistance is happening. What is the consequence of that to the bug? What is the consequence of that to the enzyme? And then using that information, can we develop new strategies for outwitting the pathogen? Can we move ahead? So we need to understand the pathogen before we address the disease. And as Rick said, I am a biologist at heart, a basic scientist at heart. So I'm actually really looking forward to learning new biology in the world's deadliest pathogens. So the pathogen is my model for transcription. And how do I do this? So I follow the advice of Richard Feynman, the Nobel Prize-winning physicist, that he said that if you want to understand a process, it's helpful to look at it. And that's what I do. And how do I do that? So I go down to the Evelyn Lipper Cryo-EM Center here, and I work with these three very talented scientists that run the center. And it's really and truly the revolution in cryo-EM methodology. So you've probably heard about cryo-EM. It's a new method for structural biology. It's the revolution in the methods and the hardware and the support of the center that we have been able to see the molecular motions of RNA polymerase. We think of RNA polymerase as a machine. It's a molecular machine. It's not a static thing. It's moving, it's breathing, and it's doing different things. And cryo-EM allows us to see that. So this is an example of what cryo-EM can do. This is the people in the lab working on purifying all the proteins here that we're going to try to understand, the imaging. And then in the end, we end up with something that looks like this, like a molecular machine. And this is from our precision instrument shop. I didn't finish last night gluing it together, but this is what it looks like, right? Really blown up, really magnified. That's what RNA polymerase looks like. And what have we discovered? So this is the RNA polymerase. This is the DNA. So the DNA has to be opened up and unwound for RNA synthesis to occur. And this is RNA starting to be synthesized, right? The intermediate molecule. It needs to go along here. This is the path of the RNA. Rifampicin, which just flashed up in orange, and it's actually an orange drug. It'll turn your tears and urine orange, will block the synthesis of RNA. It sits in the path of the RNA. So right away now we know how it's working. We know what step of the transcription cycle it's working, and that's information. Here are those other inhibitors that are not yet drugs, that are not yet in the clinical setting, and they bind all over, and they will disrupt transcription. So these are the ones. So we're trying to understand, and we have been able to understand how many of them work. And we're thinking about where do they fit in this pathway. And then this is an inhibitor. This is the only second inhibitor that targets RNA polymerase that's in a clinical setting, right? So there are only two inhibitors that target RNA polymerase. One is rifampicin, the other is fidaxomicin. So fidaxomicin sits here and it prevents the enzyme from closing down onto the DNA. So you're just seeing that movement. This is what I can see with cryo-EM, these kinds of movements. But fidaxomicin, although it's actually effective against RNA polymerase from TB, it's not used to treat TB because it doesn't get past the gut. So it's actually used to treat another pathogen, Clostridium difficile, which someone had asked me, what about hospital-acquired infections? So that's one of those infections. You go into the hospital, you're prepping for surgery, they give you antibiotics, and one of the unfortunate effects is that it'll clear out your commensals, your good bacteria, and the spores of Clostridium, if it's in the air, can actually colonize and cause this disease. The bacteria will then germinate and grow. And fidaxomicin is really good. It's a very effective treatment, and that's because it'll only kill a certain class of bacteria, so it'll allow the commensals to grow. So I'm really interested in seeing if we can get something like this for TB, growing something that's narrow spectrum that will only kill the ones I want to kill. I told you I'm really interested in understanding how does the RNA polymerase know to start making these pumps to pump out the ribosome inhibitors. So we're studying a factor called the YB factors and how it pulls the polymerases and increases, turns on that the transcription of those genes. And then this is just a movie just showing you the kind of information we can see. We can see the RNA polymerase closing in, opening up, unwinding the DNA. So we are actually learning all the events the polymerase has to undergo to start transcription. And then lastly, because this ties into the story that I want to tell you, is that all of these studies lead here right now, actually where we are. So I am learning about specific mechanisms of resistance. So one of the things we've seen, again, with cryo-EM are these movements. And what we've learned is that rifampicin resistance occurs in the target. It occurs in the RNA polymerase that the molecule has to bind. And I have a little thing just for you guys to see what it looks like. So this would be rifampicin. If anybody wants to hold it, they can. This little molecule goes in the inside of this protein, and it just stops, it clogs it up. Someone made the analogy of sands in a gear. So we are trying to understand how that resistance, what is the consequence of that resistance on the bacteria. The consequence is you've just affected an enzyme that's critical for the bacteria to grow, critical for it to survive, and therefore the bacteria are actually less fit. They don't do as well. It's not ideal. And so they acquire new mutations that compensate, and some of our studies have allowed us to understand these mutations. And a big deal in the era of information here and sequencing is that we have clinical genetics. We have the sequences of about 50,000 patients who have been treated with drugs, and we know what mutations are occurring. So we're really trying to understand all of these movements in the context of these mutations. And this information is ammunition to fight TB. So I'm going to end on the TB bit here by just letting you know that I'm really lucky. I have a physician scientist in the lab. As I said, I'm a Ph.D., so she's a real MD. But she's working with the Rock Lab and Venetia Govinda. He's another HOL here at Rockefeller that also studies TB. And Venetia is from South Africa, so she's actually personally impacted by TB itself. But they're working together to use this information to design new strategies. And the way we've done this is, this is transcription. If you look in the textbooks, this is what it looks like. But in reality, there are multiple steps. The RNA polymerase has to find the start of a gene. It has to unwind, open up the DNA, and then it starts to make the RNA, and then eventually releases the RNA. So rifampicin works at the first step here. So our idea was that if we target another step, and we target it really cleverly based on what we know about the mutations, what happens? And what actually happened is that we now have a new class of compounds if you treat the bacteria with 2 antibiotics, and this is early stages, right? This is just in the bacteria. It will kill rifampicin-resistant TB 10 times more powerfully. So that's that has so much potential for thinking about the people who have drug-resistant TB, where the mortality rate is 50%. It also works with rifampicin, the new class of compounds we have, to more potently kill I have actively replicating bacteria, so that would be active TB, but it also reduces rifampicin resistance. So that's great because that means I get to keep 2 of my compounds as a potential treatment. And then I think, which is the most exciting one here, is synergizes with rifampicin to kill non-replicating latent TB. That's really hard to do. So we don't have a lot of drugs that will actually be effective for latent TB. and rifampicin being one of them. So we're really excited to move this forward. We're sharing our data with the TB Drug Alliance to strategize these approaches, and we're hoping, they'll use this information to think about how they're designing their drugs. And the next frontier for TB is I'd like to know what the state of transcription is in situ, maybe more ambitiously in the lung. So you can actually now, the new technologies will actually allow you to see these molecular machines in the cell. So I'm no longer purifying the proteins and adding the antibiotic. I can add it to the cell. I can ask what's happening here. But this is in the future. So I'm going to quickly switch to telling you just briefly about coronaviruses. We also study coronaviruses in the lab. Like many places, Rockefeller had the initiative that you can work during the pandemic if you were addressing COVID-19 science. So, again, I'm really grateful. Again, Rick, actually, we wrote the email to Rick and asked him, can we work on coronaviruses? And this work is really a collaboration of many labs at Rockefeller, but importantly with Seth Dars and with Charlie Rice. And so unlike TB, COVID was and is an indiscriminate killer. It killed all over the world. And this is an underestimation, the 7 million, but this is what the CDC has estimated as the cost of lives. But even today, even last month, there was almost 4,000 deaths. So it's occurring all over. So it's not done. And these waves basically show you, right? These spikes, we get spikes. So we're always keeping our eye. So even though I got an emergency grant to study COVID, I think the NIH was smart enough to realize they're going to make it into a permanent grant. And so we're going to keep looking at this molecule, at this virus. So this is coronavirus. And if anybody wants to touch it later on, I have a model here. I don't know, maybe you don't want to touch it. But inside the coronavirus is an RNA genome. So it also has to do, and it has a RNA polymerase, and it has to do transcription. It has to replicate and transcribe its RNA genome. And to do that, it uses these building blocks that make up the RNA. And these building blocks actually look like this. They're very chemically different. And this is the basis of a set of antivirals called nucleoside analogs. So nucleoside analogs basically mimic the nucleosides, and they come in there and they clog up the machine. And I'm just going to give you an example. This is ATP, one of these building blocks. And this is remdesivir. You've probably heard of remdesivir, which is still an effective treatment. And it looks just like ATP, just has an extra little group here. And it hides out, yeah. And that's how it does it. It basically comes in there and just pretends it's ATP. But we're working with Gilead Sciences. We've tried to understand why does remdesivir bind instead of ATP? But also, can we now think about making remdesivir-like molecules using the other building blocks, the C, the G, the U? And that's where we are. We're also trying to understand why is this virus particularly resistant to nucleoside analogs, which is a good arsenal for treating a lot of viral infections. One of the reasons is they have a clip. It'll just clip out the nucleoside analog. So it's very effective. It's very resistant, actually, naturally to nucleoside analogs. And then lastly, we're trying to understand new targets for antivirals, right? Not just transcription, but when RNA is made in the host, it actually has to get modified in order for it to be translated into protein. It also has to be modified so it can hide out from the host immune system. And so we're looking at these processes. And then the last part of our program is brand new. It's working with Seth and Charlie again. We're going to be looking at hepatitis B, which technically I guess they don't list it as the leading cause of death as an infectious agent because it causes liver cancer, but it is a brutal killer as well too. So in conclusion, we are studying the essential processes in these viruses and bacteria, which is transcription. And we do share our data with the TB Drug Alliance and Gilead Sciences. And it's because of the generosity of the university. It's because of my great collaborators and wonderful lab, the benefactors here as well too, and our research centers that drive these discoveries. These are the people here. So if you want to corner them later on and ask them questions, they're all here. And I want to thank you. As I said, this encapsulates my program.
Thank you, Liz. Wonderful talk. We have time for questions, so please raise your hand if you have a question for Liz.
Yeah, so there are people who are allergic to penicillin, and it can cross-react. My sister was one of them, actually, and so they had to find other treatments. And that is a problem because penicillin and that class of compounds is important. But yeah, unfortunately, rifampicin is not in that class.
Thank you. Is there a question over there?
Hi, I'm Sally Permar, and I lead pediatrics at Weill Cornell, and I'm also a pediatric infectious disease doctor. So I'm glad to get to meet you and hear your talk. But I wanted to ask about TB in children is a major issue. The diagnostics is very troubling for both adults and pediatrics, but there are a lot of cases that don't get diagnosed until they're very end stage, and extrapulmonary disease is very common in children. So I was wondering, do you have any insight into the differences in the transcription that's going on in extrapulmonary disease versus latent versus active? Is that on your horizon?
Yeah, I mean, that's an excellent question, and that is exactly what I would love to study. So, for example, that's what I mentioned the in situ work, right? Can we start looking inside the cell at these large machineries? What is happening for now, we'll probably use models. So like the granuloma models or, I'd have to learn what the models are for children. But you're right, the disease in children are different, and it tends to be more extrapulmonary, and they don't get these granulomas. And yeah, no, it is the future. Oh, awesome.
Lulu?
Thank you. This has all been fascinating. And it seems like the work you're doing in discovering how to interrupt the transcription process probably has a lot of pathways to other disease approaches. You mentioned Hep B at the end. Are there other diseases that you're currently beginning to envision that your science might be able to address?
That's a great question. I mean, I focus on those three. What I discovered are applicable, right? So, for example, hopefully some of these new drugs will be used to treat the other infections, including the antivirals and the antibacterial. I will note rifampicin, although it is effective against many other infections, they reserve it for treating TB because it's one of the very few effective things we have left. So I'm hoping, discovering new drugs, maybe new diseases can be addressed. But right now, my focus is on TB and the coronaviruses as a model.
And as you get to know more about the interruption process of transcription, are there other ways to approach developing new drugs, pathways, environments, other microbes that could help you disrupt the process?
So you're asking whether there are other pathways, like biological pathways in the cells for disruption, or
learn how you disrupt the transcription process. Does that open up more pathways to other ways to approach it?
Yes, absolutely. And that's actually a really that's a very prophetic question. But that is exactly what we're learning. So we're learning transcription and translation are actually working together. And we learned that from some of the clinical genetics, but we learned that because some other bacteria might be doing the same things as well. So the next step would be the downstream one, and how do these two molecules and these two pathways coordinate? We definitely have to be thinking about how the pathways talk to each other, because that's going to make us more powerful to address this, make us more clever, more information.
Thank you.
Yeah.
Wonderful talk. I mean, it's really amazing how nicely accessible you made it for all of us. I've studied the escape pathogens, and I wonder What do you think about how your methods and study of TB can translate to other pathogens? That's question one. And question two, with all of the talk about Rosetta fold, alpha fold, and all of these folds, and there are many more methods from deep learning, computer vision that can help in not only mapping out the machinery, but also generating new ways of stopping that machinery. So what do you think about that?
Yeah, I'll address the escape pathogens first. So again, I think the approaches that we're developing will be applicable to other pathogens. The reason I like the approach is that normally, if you're taking antibiotics or a combination therapy, you're taking it usually to address different aspects, different biological pathways, right? So maybe transcription, translation, cell wall synthesis, or something like that. What we've learned here is if we went against the same pathway twice, just in a transcription, but in different steps, that it's actually more potentiating, it's more powerful, and it actually causes less resistance on the drug, the first line drug, like the one you already have. So I think this approach could be used for thinking about treating escape pathogens. Escape pathogens are going to need new antibiotics, so that whole transcription pathway offers us targets right there. Second is you're asking about alpha-fold 3. So yeah, as a structural biologist, I'm very much intrigued by alpha-fold 3. It's generated some interesting new hypotheses. One of the things I do want people to be aware of, it can be wrong. So every time it generates a hypothesis, it has to be tested. But it's great for hypothesis generation, and it's good for, maybe for culling what you're going to study. Like there's a lot of things to study. Here's, AlphaFold said, maybe these are the things that interact with RNA polymerase. I'll focus on these. But you always have to test and validate it for now. Yeah.
Allison?
Thank you. First of all, thank you, Liz. That was amazing. And I'm so glad you're working on it. Ever since I read Paul Farmer's book, Mountain Beyond Mountains, I've been really interested in the multi-drug resistant TB. But I hope you'll forgive me because there weren't other questions. I want to just ask a little off track. And that's also personal experience. So for the last year, I was treated for disseminated histoplasmosis. Rick knows because he visited me in hospital the day they diagnosed me at Weill Cornell Infectious Diseases. So I caught this because I was on Humira. And therefore, it took hold of me, and it's fungus, obviously. And so I became acutely aware that there are only about 2 antifungal treatments at all in the world, and very few people are working on this. And what's fascinating is that the doctor who treated me said I was the first case he'd seen in 10 years. When I last spoke to him, he'd seen 4 more in a month. So fungal infections are so on the rise. So I'm just wondering, do any of your things that you're working on, do they also impact fungus, or is it just such a completely different pathway that it's not possible?
Yeah, I think because they're so different, and I think one of the reasons, and there are MDs here, so correct me if I'm wrong, one of the reasons it's so difficult to treat fungal infections is because their enzymes are more related to humans' enzymes, so they're very toxic and they're very hard to really take a lot of antibiotics or antifungals then. It's not something I'm working on, not yet. I'm 54 years old, maybe in 10 years. I don't know. We'll see.
There's a question here.
This is fascinating. Thank you. So fighting these and other diseases is a race against time to save lives. How do you leverage technology and artificial intelligence to really shorten the process, get faster to that cure, that discovery that can really save lives?
Yes. So I don't know if you're aware, we actually have an artificial intelligence lab here at Rockefeller, or artificial intelligence group I'm actually engaging with them to ask just that question. So for example, how can I start to think about the human side of things, like the human proteins and how they interact with all of these, and start to really understand. Again, my goal is just to really understand it better and then maybe make predictions. But I think we are going to start to use AlphaFold 3 and other machine learning approaches to address just that. Because I think we have an opportunity to be broader, right? To think more broadly than just the enzyme. And so I'm excited to start doing that. Thank you.
So I'll ask a question in the absence, and others think about what other questions you want to ask. So one of the real issues in the treatment of TB is the need for the duration, 6 months to up to several years. You know, one of the things that made GLP-1 successful was going from a drug that had a half-life of a couple of minutes to one that has a half-life of a week. And it seems like it would be transformative to TB if you had very long-acting drugs that rather than trying to get people to take so many pills every day, which is very hard to do, you might get to, well, you only need to get perhaps a subcutaneous injection once a week, and that would be much more feasible, it would seem, for adherence. Is that possible for small molecule drugs, or is that going to be limited to peptides, or how do you think about that?
I don't know. I mean, that is a big bottleneck for us. The stability of the drug, in general, it does. The liver just takes everything out. That would be I mean, I would say the TB Drug Alliance would have to take it from there. So I collaborate with a medicinal chemist. That would be something they can look into, seeing if they can increase. I mean, I'm trying to think. Yeah, that would be ideal if it didn't.
An alternative would be to try to break down the granulomas, which is this protective environment for the bugs. Are there efforts in that? I know that's different from what you're doing, but in the field, is that something that people are actively working on?
I don't think so. I don't know. I mean, I think a lot of the focus has been on treating active TB right now because that's what's killing patients. As I said, I'm really interested in looking at latent TB to really eradicating it. You're talking about physically breaking up the granule?
That's the protective environment, right?
Yeah. This bug is something else. Yeah, it's hiding in the very cells that's supposed to be clearing the infection. I don't know of any such study.
Yes, here.
Is your study... Great talk. Is any of this translatable to vaccines to prevent the 3 diseases?
To prevent TB?
Otherwise, yes.
Oh, yes.
Well, or more specifically, COVID and Hep B.
Oh, I see what you're asking. Yeah, so for the TB stuff, there are Hep B vaccines as well too. But unfortunately, I think people with Hep B already have Hep B, and they pass it on to like their offspring and stuff. So the Hep B vaccines work. So for new, right, Charlie, for newly acquired infections, it's the people that already have it that are still dealing with it and passing it off. The work I do might not necessarily affect vaccine development. I'm thinking about it from the other approach. Ideally, vaccine is great, right? Because that'll bypass, because of vaccine hesitancy or maybe people not being able to take the vaccine, we always want to have that arsenal of antivirals and antimicrobial. And there is a vaccine for TB. It's usually given, I've had it, right, because I grew up in South America. It's called the BCG vaccine. It's an attenuated strain of related. But it's not that protective, to be honest. It doesn't work that well other than make you become a red flag whenever you go anywhere and get tested. You might have TB. So it's effective, I think, with children, young children. There's some efficacy there. But we don't have a real... I know what's really interesting is that a TB can infect, and what you think maybe you've cured TB, and then it can reinfect. So I'm really worried about the vaccine efforts there. I don't know how it does that, to be honest.
Well, I know we could go on for a long time, but I see we're at the hour, so please join me in thanking Liz once again for a great talk.